Chapter 15. How are genes inherited? People sometimes look the same or similar to their parents, and that is because they share some of their genes. So really, genes are passed on from one person to the next, and so we always shuffle some genes around. And now let's look at the inheritance of genetics. Let's first start with the structure of the genome. The genome itself is basically a term that describes the whole genetic information of a person, and that is 3.2 billion letters, and genes interspersed, they only make up about 1% of the whole genetic code. We have around 30,000 such genes. And as I described in What Are Genes in the other chapter, the human body has a nucleus which contains the chromosomes, and the chromosomes contain the DNA genetic letters. The chromosomes are, you can say they're kind of like boxes, and they contain around 1,000 genes per chromosome. There are different types of chromosomes. So if you list them all up, you always see that you have 2 copies of every chromosome. There's chromosome number 1, chromosome number 2. They all have different sizes. And as I said, each one of them will have around 1,000 different genes on there. And then we have the X and the Y chromosome, which we're going to talk about in a minute. Now, I've colored them pink and blue because the pink ones are all from mom, and the blue ones are from your father. So that is why we have 2 copies of every chromosome. You get 1 from your father, 1 from your mother, and that is why you kind of have your mother's eyes and your father's smile. So really they're shuffling their genes to create a new combination, which then is you. As I said, 1,000 different genes per chromosome. And for example, the lactase gene for lactose intolerance sits on chromosome number 2. The factor V gene for thrombosis sits on chromosome number 1. And one example that I've made in the other chapter, COL4A5, sits on the X chromosome. So different locations for different genes. And now let's look at heredity. Let's first ask with a simple question. What determines a person's gender? Is the person male or female? And this is entirely defined by the X or the Y chromosome. You might have heard about this. Having an X chromosome and a Y chromosome means you are a man, a male. And if you have 2 X chromosomes, you're female, a girl. So let's look at a couple that together have a child. The man has an X and a Y because that's why he's a man. And the woman has 2 X chromosomes. Now, when they have a child, they will by chance pass on one of the 2. So the man can pass on the X chromosome, and from mom, the baby will also get an X chromosome. So 2 X chromosomes, it's a girl. If the man produces a sperm that has a Y chromosome, so with a 50-50 chance, then the baby will get 1 Y from the father and 1 X from the mother, will be a boy. And again, XX is a girl and XY will be a boy. So really what defines if it's a boy or a girl is the chromosome that the father passes on. And you remember, Middle Ages, often kings blamed their wives for not giving them sons. It's really the sperms of the king or the father that were the cause. So really just by knowing which of the chromosomes have been passed on, you know if it's a girl or a boy. And this is the basic mode of inheritance, and this is also how it works with other genetic traits and diseases. If we look at the factor V gene on chromosome 1, as you know, as I said, you get 1 chromosome with the gene from your mom and 1 from your father. And now I need to teach you some technical words. There's codominant inheritance, which means if you get 1 genetic defect in 1 gene, you have an increased risk. And if you get 2 of these genetic defects, you have more risk. So the more genetic variations you have, the higher your risk. It's a kind of logical understanding. So let's look at 2 people, and they both have 2 copies of the chromosome. So the factor V gene on chromosome 1, the woman is going to have 2, 1 from mom, 1 from dad, and the man is going to have the same. And now let's mutate one of these genes in each of those people. And we know from scientific studies, if one of your two genes is broken, then you have an 8 times risk of developing thrombosis compared to people with no broken genes. Now, again, just like with the X and the Y chromosome, they're going to pass on one chromosome by chance. So if this person, in this case, it's the healthy chromosome or the healthy gene passed on, this child will have a normal risk of osteoporosis, of thrombosis. In this case, the baby inherited one broken gene from mom, but a working gene from dad. It's just the same as the parents, risk times 8. Here again, risk times 8. And this baby is unlucky, it inherited the broken gene from both parents. And here it's actually not 2 times 8, 16-fold, but 80-fold. So really having more risks together multiply by each other. So it's not additive, but multiplicative. So by having two parents which have an 8-fold risk of developing thrombosis, you can get anything from no risk to 80-fold risk, 10 times higher than the parents. And this is, as I said, is codominant inheritance. The more genetic defects or genetic variations you have, the higher your risk. Now the question, if you said, why do a genetic test? Let's just look at my family history. If you assume that you are the same as your parents, this baby is gonna have made the wrong conclusion. It has an 8-fold lower risk than its parents. Those two will be correct. They have the same risk as the parents, and this child has a 10 times higher risk. So really, by assuming you're the same as your parents, you have a 50% error rate. So how can we really find out? We can test the gene, and then we can find out, did you inherit any of it? Did you maybe inherit more of it than your parents? And so you can get more detailed information. So this is codominant inheritance. The more genetic variations you have, the more risk you have. Now there's something else called recessive inheritance. And that means you need 2 broken genes for this disease to occur. And let's look at another example. Again, we have 2 people. They have 2 copies of the lactase gene. That's the one that digests lactose, the one for lactose intolerance. And now let's make 2 of these genes lactose intolerant, so mutated. What happens if you have one gene that does not know how to digest lactose, but the other one can? It's enough for the body to know how to digest lactose, because this gene can take over the function for both. So really, people, they're carriers, we call them. They have one genetic variation which disables it, the other one works, and they are lactose tolerant. They can drink milk. And this person can drink milk, has no problems. And the father, again, we give them one defective gene and one functional one. Also, this person is not lactose intolerant. Now they have children together. Again, by chance, they pass on one of the 2 chromosomes or genes. And this child has received 2 healthy genes. This child has received 1 mutated gene. This also, but from the other parent. So these are carriers, but they don't exhibit lactose intolerance. But the 4th child has received 2 broken genes, and both of them don't know how to digest lactose. So this child is going to be lactose intolerant. So again, looking at the family history, why do a genetic defect? Let's just look, are my parents lactose intolerant? If the answer is no, you would say, well, then it's, I'm not going to be lactose intolerant. Is incorrect. Because this child is lactose intolerant. It got it from its parents' genes, but the child could not see this in its family history. So this is recessive inheritance. Again, a genetic test is going to tell you. A family history tree is not going to tell you. And then there's dominant inheritance. So we had codominant, the more genetic variations, the higher the risk. Recessive, you need 2 broken genes for the disease to occur. Or dominant, which is one of them is enough to give you the full risk. And again, let's look at a gene. Every parent has 2 copies. The HLA-B27 gene. It is a genetic variation that increases the risk of Bechterews disease, which is basically a bone disease the invertebral column begins to fuse together. So instead of having the flexible invertebral discs, it becomes one solid bone, and then people start to get posture problems. They can't move the spine anymore. And this is very strongly influenced by a genetic variation. Now let's give both of these people this genetic variation. They have a 78-fold risk of developing the disease, and more than 95% of all people that have the disease have this genetic variation. Now, both of them have high risk. Now, they by chance pass on one of the chromosomes, and this child is going to be healthy. Even though both parents have a high risk, this child has no risk. And if one of them is passed on, 78-fold risk, as you would expect. However, if you get two of the genetic variations, the same risk again. So really having more of the same genetic mutation does not increase your risk to be higher. And this is dominant inheritance. So one genetic variation is enough for you to get the full risk. And again, you get a 25% error rate when you look at the family history compared to a genetic test. And that's the end of chapter 15, how genes are inherited.